introduction. Are we good to start? Okay. All right, then let me go ahead and share my screen. Okay. All right, well, thank you, Dr. Kisanchi, for the amazing introduction. And again, thank you to the organizers for having us here today. It's great to get a chance to talk about some of the results that we were able to get the last few years. So, like Dr. Kisanchi had said, this is a combined effort between both me and Dr. Molnar, Sebastian. So, in the beginning, I will be talking about more of the monocyte derived EVs and HIV infection. And then in the second part, Sebastian's going to be focusing more on the T cells. So, thank you all again. So, I don't really have to belabor this too much. I know this is an HIV heavy group, but of course HIV or human immunodeficiency virus is a worldwide epidemic that's still impacting around 40 million people within the world. And we're getting around 32,000 new infections, at least in the US yearly. So still something that is continuing to be a problem. And of course, left untreated, it leads to acquired immunodeficiency syndrome, or AIDS, which is characterized by the depletion of CD4 T cells. And this allows for opportunistic infections that can ultimately lead to death. So a little bit about the virus. As we know, it's an enveloped spherical particle with the virology textbook saying it's around 100 nanometers in size. Some of the important proteins we'll be looking at today are accessory protein NEF and GP120, that's our surface marker. And then the genome for our HIV is a single-stranded RNA positive sense virus. And some of the RNAs we'll be looking at today are going to be our TAR, which is in the 5' long terminal repeat area, and then our envelope or more of our full-length genome RNA. So for our viral life cycle, we have HIV that comes and attaches with our CD4 receptor and either the co-receptor CCR5 or CXCR4, fuses with the membrane of the host cell, which releases the capsid, allows for the reverse transcription and pre-integration complex getting into the host nucleus, where it is then able to integrate within our genome. Once it's in the genome, it uses the host transcription and translation machinery to produce viral RNA and protein, which are eventually assembled out into new budding viruses where they mature and continue the cycle. Although there is no cure for HIV, there is our therapeutic regime or combined antiretroviral therapy, CART, that targets multiple stages within the viral life cycle. And the introduction of CART has expanded or extended the lives of those living with HIV to similar lifespans of those without. With these extended lifespans, they're, although of course great to have, again, the prevention of AIDS, but there are still a variety of complications associated with HIV, and these stem from no transcription and no translation and inhibition. Because since it uses the host machinery, it's hard to target just that HIV virus part of the transcription. So even in latent cells that are on the CART, you still have leaky transcription and leaky transcripts of viral RNAs produced that lead to toxicity within the surrounding environment of that cell and can lead to things like chronic inflammation. With these HIV-associated complications, the one that I am most focused on is HIV-associated neurocognitive disorder. And this encompasses around 50% of people living with HIV develop HAND. And with the treatment of CART, we've gone from the most severe form, which is our HIV-associated dementia, to the less severe forms, including the asymptomatic form. However, even with this asymptomatic form of hand, it still impacts the lives of those living with hand, reducing memory, can contribute to depression, and they can have issues with impairment of executive function. So the people living with HIV, as we're getting a larger, older group, now with our CR that is extending life, this is going to continue to be a problem that is needs to be addressed. And so some of the contributing factors for hand is the neuroinflammation. So one of the now recognized reservoirs of HIV is the microglia within the brain. And so the microglia within the brain are producing these low-level viral transcripts and causing inflammation within the microglia and the surrounding astrocytes. These can eventually lead to some neuronal loss, interruptions within the transmitters between the neurons, and as it continues, can lead to an abundance of amyloid plaques and tau depositions, which can be, of course, associated with that HIV-associated dementia. So some of the goals that I had while doing my graduate school was for looking for a therapeutic to help combat hand. And one area of interest is cannabinoids, and these are derivatives from the cannabis plant. The reason that this really started was because there were some papers like this one by Watson that showed the cannabis exposure reducing the likelihood of neurocognitive impairment in people living with HIV. So their lab, as well as others, have continued to look at some of these components within cannabis, including cannabidiol or CBD, cannabidiol, and CBD, which in our hands had previously been shown to help reduce some of the effects of the HIV infection and reduce viral transcripts as well as viral proteins. And then in my study, we look at a similar structure to CBD HU308, and that's what I'll be talking about today. So like I said, our CBD has a similar synthetic analog, HU308. The reason HU308 was tested was for its ability to target the CB2 receptor specifically. It's an agonist for our CB2 receptor, where CBD has a variety of receptors that it interacts with that allows for it to have multiple pathways affected. So we wanted something that was a little bit more specific for the anti-inflammatory pathway. So one of our first activities was just to see that if it does still impact the intracellular viral levels of RNA. So here we have uninfected monocytes, or THP1, and then our infected monocytes, THP89, and these are treated either with CBD or HU308. And in our hands, what we saw was that with either CBD or HU308 treatment, there is a decrease in our viral transcripts of TAR, TARGAG, and ONF. And then what was interesting within this study is we took those same cells, and these are chronically infected monocytes, and we activated them with LPS and ATP to see if, for example, adherence to CR was not kept, and you had that activation of your viral transcription. Are these components still able to reduce those viral transcripts? And so our latent are these lighter color, and these darker color versions are the ones that are activated, or the ones treated also with LPS and ATP. And what you can see is in both cases for our latent and activated versions of all three transcripts, both CBD and HU308 are able to decrease these transcripts. Furthermore, it was interesting that our HU308 specifically did a greater decrease in the activation of these transcripts, potentially indicating a unique method to it compared to CBD. Similarly, we saw a similar trend in our primary macrophage infection. So here we had PBMCs that were infected with a dual tropic 89.6 HIV virus. These virally infected cells were then treated with our CR treatment, CBD or HU308. And the treatment for this one continued on 3 days. At the end of those 3 days, we looked at those viral transcripts and saw, even in our primary macrophages, a decrease in viral transcripts in both our HU308 and our CBD compared to the infected control. Not just with viral transcripts, but we wanted to see if this is actually impacting the proteins as well. Again, the proteins can be released and affect neighboring cells. And what we see is with either CBD or HU308 in increasing amounts, there is a decrease in viral NEF within the cells. And these ones are chronically infected monocytes or U1 that were treated with either CBD or HU308. And in both instances, again, a decrease in NEF and then a decrease in NF-kappa B phosphorylation, which is associated with our viral transcription. So overall, intracellularly, we are seeing that these cannabinoids, CBD and HU3, do decrease viral transcripts in both latent and activated cells or monocytes, and that they also decrease viral proteins such as NEF within those infected cells. But we really wanted to get to the bottom of how do these infected cells, again, they are mostly the microglia that are holding these infections within the brain, or a major reservoir. So we really wanted to get to how do they then impact the surrounding neurons and see that neuronal death that's associated with ANT. So in order to answer this, we looked at extracellular vesicles, which are lipid membrane-bound vesicles or particles that range from anywhere of 50 nanomolars all the way up to 5 micromolars. Extracellular vesicles, or EVs, range in size. They have differences in their biogenesis. Some of them are budding out. Some of them use the escort pathway. They vary in function. Almost all cells that have been tested have shown to have EVs as a very important form of intracellular communication. Even bacteria cells are now being shown to also have a form of EVs. And what they're carrying are components of the host cell. So there's lipids, proteins, and nucleic acids. But in the case of infection as viral infection with HIV, it also carries pieces of either proteins or mRNAs or long non-coding RNAs from the infected cells to neighboring and distant cells, which can contribute to disease pathogenesis. So we wanted to take a look at the EVs that were being released by our HIV-1 infected cells and compare them to the treatments that they were receiving with either the CBD or the HU308. And so to look at the different populations of EVs, we use differential ultracentrifugation, in which you have your cells, in this case, HIV-1 infected monocytes, grown in large volumes up to 25 mLs, and spun down at 500 G to get our cells. The supernatant from those cells were then transferred to another vial, spun at a higher 2000 Gs, this now brings down our first, this pellet now brings down our first extracellular vesicle population, or what we're going to call 2K, based on the spin speed that they were collected at. And these 2K populations are going to have a variety of the larger extracellular vesicles, including our autophagosomes. The supernatant from those 2K were continued to spin down at a higher speed of 10K. Similarly, that supernatant was spun at 100K, the following at 167, and the last one at an additional 167, but for a longer time, or 16 hours. Each of these populations have different subpopulations within them that make up the majority of the pellet. And really for my talk, the ones that I'm going to have the most interest in are these 100K, or the classical exosomes that go through the S-Kurt pathway. And then the 2K, which are more of the autophagosomes. So these ones are considered to be typically budding out. These are our larger EVs that bud out from the host site. So the first thing I did after collecting these EV pellets was to analyze them for their concentration. And here, throughout the next few slides, I'll use the same color scheme, where our blue is going to represent our HIV infected cells. Our orange are going to be infected cells that were treated with CBD over 3 days. And then our yellow are going to be our cells treated with HU308 over those 3 days. And for our different EV populations, what we can see are both our CBD and HU308 decrease the classical exosome population, or the 100K population, without significantly changing their EV size. And this is consistent with results that we have seen previously published in our lab, as well as others. These EVs were then isolated for their RNA, and I've used an oligo-DT to determine, as the RT step, to determine mRNA of IL-1 beta and TNF alpha. And in this case, we saw a decrease in the cytokine, pro-inflammatory cytokine mRNA within the 2K. So seeing a difference in the packaging or release of the EVs in the 100K versus the pro-inflammatory cytokine reduction, which is actually within the larger 2K population. The viral RNA was also observed, and what we could see is that with our TAR and ON for our CBD treatment, the decrease was in that 100K, or the classical exosome. And similarly for our HU308, there was a decrease in the TAR and ON in the 100K. Taken together, this is really showing that with either CBD or HU308 treatment, our chronically infected cells are reducing the 100K, or the classical exosome population release. And then within that 100K population, there's also a decrease in viral transcripts. And then interestingly, a decrease in our pro-inflammatory cytokine RNA, our TNF alpha and IL-1 beta, within the larger 2K population. We next wanted to have a more accurate model of what infection looks like in the brain. So here we used neurospheres as a central nervous system model. And for this work, this is really going from Dr. Heather Branscombe, who is within our lab and has helped with our neurospheres. She took somatic cells, and through a variety of different reprogramming steps, pushed them towards a neurosphere configuration. These neurospheres containing our neural progenitor cells, our neurons, astrocyte and microglia-like cells, which you can see in these images here with our neurons shown in this merged pink along closer towards the outside, as well as our astrocytes and our microglia-like cells. Not only did we want to make sure that these neurospheres had the cells associated with HIV infection of our microglia and our astrocytes, but we also wanted to make sure that they could be, in fact, infected with HIV. So our neurospheres were infected with our dual tropic 89.6 virus for a week, and then treated with CR. And so for our untreated ones, we can see viral PR55, P24, and NEF. And then once you add that CR treatment, you can see a reduction in viral PR55, 24, and NEF. And then finally, what you can see here are that the neurospheres, and these are 3 separate neurospheres imaged in each of these pictures, that they are still observing that nice neurosphere round structure, even all the way up to 14 weeks. Okay, so once we knew that we had a neurosphere system that worked, what we wanted to do was to treat them with these cannabinoids. So here we did a pre-treatment of cannabinoids 24 hours prior to our HIV-1 infection with 89.6, and then continued their treatments every other day for about a week. At the end of the week, we took those cells, or the neurospheres, isolated the cells and isolated the supernatant to look at the EVs compared to the cells. And what you can see here are our neurospheres that were infected with HIV did in fact have our viral transcripts TAR, TARGAG, and ONV. As expected, our CR did decrease those viral transcripts. And then with either CBD or HU308, there was still a decrease in those viral transcripts compared to the infected alone. And for the extracellular component, like I said, here we use the supernatant from the neurospheres and isolated the RNA, and similarly saw a statistical decrease in all treatments, so our CR alone, our CBD and CR, and our HU308 and CR. We next wanted to take this to a humanized mouse model. So here we used an NSG humanized mouse model, in which was again infected with that 89.6 dual tropic strain. With this infection, we treated with CR, and in this case, we had 2 different versions of CR. We had a CR in a low dose, which is 1/10 the recommended dose, and then the normal or optimal dose of CR. And then we treated with HU308 by itself or with that lower dose of CR. And the intention here was eventually, when we get this through 2 clinical trials, our intention is to have these cannabinoids in combination with the CR treatment that is there today, and hopefully in a way that can reduce some of those more toxic side effects that we see with the cannabinoids. So when we look at the results, what we see, of course, in our infected mice, we have viral TAR, TARGAG, and ONV transcripts. Our low or 1/10 dose of CR decreases the TAR and ONV, but only with 1 star. But then our CR, that's at the optimal dose, was able to now bring these down closer to 2 logs less within the mice. And for here, each of these were a mice N equals 3 for each of these treatments. And then our HU308 alone was also able to bring our viral transcripts down, similar to that of the CR optimal dose. But really, I think the most exciting data from here is that our CR that was at the low dose combined with HU308 brought the viral transcripts down at a significant level that was even significant to that optimal CR alone. So really showing here that that idea of that combination between the viral or our typical CR treatment along with HU308 or other cannabinoids do potentially have the ability to decrease these viral transcripts. And hopefully in the future, we will have more studies that also show more of that pro-inflammatory effects of these viral infections. So what we have proposed here is with our HIV infection, our monocytes or myeloid cells have viral accumulation that occurs within them and have these EVs that are released. These EVs are carrying viral TAR, NEF, and IL-1 beta, which can go on to recipient cells that can be our neurons or astrocytes or other cells within the body, increase cell death, and in the case of hand, increase more of the symptoms that we attribute to hand. And we're hoping that these cannabinoids, both CBD or HU308, can help to reverse this by decreasing the viral RNA within the myeloids, decreasing the EVs that are being released from the myeloids, and then decreasing some of those harmful viral components in the EVs themselves to prevent that cell death and symptoms associated with hand. So with that, I briefly want to just thank everybody. I know Sebastian's about to go, but thank you guys again for hosting me. I'll take questions, I think, at the very end after Sebastian, just to keep everything running. And of course, thank you to everybody in my lab and my collaborators. Okay, and let me stop sharing. And Sebastian, it is all you.
Hey everyone, so my name is Sebastian, and today I will talk about methods that identify different particles that are released from infected T cells. And basically we have published this work in the Journal of Extracellular Vesicles last year, so I will just present a couple of highlights from this study. Our study goal was to ask the question that would address that T cells can HIV-infected T cells various sizes of extracellular particles, and if so, what are their unique characteristics? And during this process, we had few objectives, some of them regarding to size stratify and characterize extracellular particles released from the infected T cells to look at their biophysical, biochemical, functional properties, try to interfere with the HIV infection cycle, look at their single extracellular particle immunophenotyping, look at alternative isolation and purification methods, and also recently, this is more like recent developments, we looked at their in vivo functionality. And the cell line model we used, it was the J1.1 LEV HIV-infected T cell line, which is chronically infected with the subtype B virus. Our method of isolation, the first method of isolation was differential ultracentrifugation, and we can see in this panel A, where we increased incrementally the speed and the time to obtain smaller and smaller extracellular particles, with the last spin being, how do we put this? Where is it? With the last spin being for 18 hours, 167,000 G force to obtain particles in the size range of exomeres, and these particles are in fraction E. So from now on, I will refer to them as fraction A through fraction E, which represent different subpopulation of extracellular particles. So first we did their sizing, where we used NTA, nanoparticle tracking analysis, which we also confirmed by transmission electron microscopy. And what we observed that extracellular particles fraction A to fraction D had a peak size around 130 nanometers, while the 167,000 G4 spin for 18-hour fraction E with orange color had a peak size of 18 nanometers, which we also confirmed by electron microscopy. More details we can find in the paper that we recently published. Next, we looked at the biochemical and functional characteristics, and what we observed that fraction C and fraction E contain majority of the viral RNAs as Anastasia detailed them, TAR, TARGAG, and ONV, including in fraction E, where we had a very high peak for these viral RNAs. Then we looked by Western blot for host and viral proteins. We looked at the cell line specific protein CD4A5, indicating that the EVs or the extracellular particles come from a T cell. And then what we observed that the small extracellular particles in fraction E were enriched in CD63, CD9, and HSP70. When we looked at the viral proteins, we observed the presence of GP 160, 120, various molecular weights for NEF and GAG in fractions A, C, and E, with a single P24 band in fraction C, indicating that the GAG in that respective fraction, its protease processed, just as it was detailed before in the literature. And then, as functionality, we infected MOD4 T cells, and we observed that all the fractions contained infectious virus. To our surprise, even fraction E, indicating for the first time the presence of a small HIV under 50 nanometers. But also we observed that fraction A had infectious virus as well. And then we asked the question, can we block the entry of the virus in the target cells? To this extent, we used HIV-specific monoclonal antibodies that target different regions of the envelope surface glycoprotein at the AMPA region, at the CD4 binding site, and then the top, the V1, V2 region, which is present only when the trimer is fully formed. And what we observed that the GP4E1 AMPA-specific antibody failed to block it greater than 50% the infectivity of the virus in fractions A through D, but successfully inhibited the infectivity of the virus in fraction of the small virus. The GP120 binding site was VRC01 blocked across all the fractions, the infectivity, and PG16, the trimer, failed to block the infectivity of the virus in fraction A. So this also indicates that there should be a membrane and should be a membrane that the trimer is being attached to, and also that it's an envelope-mediated entry of the virus in respective fractions. Additionally, we interfere with the virus production by treating the infected cells with a combination of NRTIs. And we observed that when the producer cells were treated with NRTIs, the infectivity of the virus in fractions A through D was greatly reduced, but the infectivity of the virus in fraction E stayed the same, indicating that perhaps this virus has other properties as well in the respective fraction. Then, as another highlight of this paper, we did single particle immunophenotyping, where we probed for virus and host cell specific proteins. For virus, we probe for integrase, which is centrally located, and also for GP120, with GP120 antibodies, VRC01 and PG16, and for tetraspanins, we probed for CD9 and CD63. And here are two representative images on the top of fraction C, on the bottom of fraction E, where we can appreciate the co-localization of these markers also indicates size differences as well. And also that the integrase with red color is centrally located, and the tetraspanins and GP120 proteins all spread around in a concentrical fashion. When we looked at the statistical analysis of the results in panel A, we observed that the virus in fraction C was predominantly CD9 positive with red color, and the virus in fraction E was predominantly CD63 positive. Furthermore, we immunodepleted the fraction E with anti-CD63, which greatly reduced the infectivity of the virus in fraction C, indicating that indeed a small virus in fraction E it's CD63 positive. Another orthogonal method to isolate the small virus was by serodifferential filtration, where we did 2 depth filtration steps followed by 2 trackage filtration steps, and we kept the supernatants and we concentrated them. First, as we did before, we sized them and by NTA and also by TEM, where the results indicated that in the less than 50 nanometer filtration steps, we had a peak size of 28 nanometer. In the less than 450, we had a peak size of 128 nanometers, and it had a broad shoulder to the left, indicating that we also have smaller particles in the filtrate of less than 450, as we can see it in this transmission electron micrograph. Additionally, we did infectivity, which indicated that the virus that passed through the less than 50 nanometer filter is still infectious. Furthermore, we infected PBMCs, peripheral blood mononuclear cells, with a ball virus. And we did the serodifferential filtration, we did the sizing, and we did the infectivity, which indicated that the less than 50 nanometer virus was still infectious at day 4, day 7, and day 14, even though the less than 450 was more infectious. This is due because it contains both the small and the classic size virus as well. And we also infected the cells with the LAV virus, where we had similar results. Here, the peak for the virus that went to the 50 nanometer was around 16 nanometers, but the infectivity profiles were virtually similar. And now let's switch gears. Let's go back to the large fraction. This is the very first fraction in the serodifferential ultracentrifugation, which goes through a 2000 G-force spin for 20 minutes, and this is what we called fraction A. That fraction was put onto a 70 nanometer ISON column, and it was divided in 40 subfractions, which were pulled into 8 pools of 1 to 5 fractions as they come off the column. And the Western blot data indicated that the very first fractions 1 to 5 were positive for amphisome and viral markers, which when we looked individually at the respective fraction coming off the column, we observed that if fraction 1 and fraction 2, and these are the very large ones that they come off first the column, contain GP120, NEF, P20, and P24 markers, indicating that the virus sits in that very large amphisome-like vesicles. Furthermore, this large fraction was immunoprecipitated with Rep7, Rep5, LAMP1, and LC313 amphisome markers. And in panel A, we observed that majority of the GP120, P24, and NEF was in the Rep5 and LAMP1 positive vesicles. And then when we did QRT-PCR for TAR, TARGAG, and OMV, we observed that the LAMP1 positive vesicles contain majority of the virus. So thus far, we have 3 different fractions that contain majority of the virus, the very large one, fraction A, the medium-sized fraction C, which is about 130 nanometers, and the very small one, fraction E, which are less than 50 nanometers. So next, we ask the question, what is the in vivo infectivity of this virus? And to this extent, we infected NHG mice, 6 NHG mice, and we followed them longitudinally. At day 94 and 115, we executed blood draws to monitor for the virus in the periphery. And then we followed the animals where we observed that the median survival for the animals in fraction A was at 217 days. The median survival for fraction C animals was in 212 days. And the median survival for the small one showed us at 193 days. The blood viral RNA indicated that at day 94, majority of the TAR, TARGAG, and on-viral RNA was the highest in the animals infected with the fraction E extracellular particles, followed by C and A. By day 115, This went down, and then the fraction C actually took over. Furthermore, when we looked at the tissue RNA postmortem in the animals, we observed that when we compared the fraction A infectivity of fraction C, it was a significantly higher tissue distribution of the viral RNA in fraction C in lymph nodes, spleen, liver, lung, and brain was actually a little bit less. When we compared fraction A to fraction E, all of them were higher in the animals infected with fraction E. And then when we compared fraction C and fraction E, there was no significant difference in the pairwise comparison. However, the trends indicate that in the lymph node, spleen, and brain, and even in the liver, there is a greater amount of viral RNA that in the animals that they were infected with fraction E. And in collaboration with Florida International University with Nazira El-Haggis Laboratory, we did a transepithelial, transendothelial resistance experiment where brain microvascular endothelial cells were infected or were treated with fraction A, C, and E, and the resistance between the two, the transfer experiment was measured, which indicated that on day 1, the wells treated with fraction A and C had a reduced TER resistance, indicating that basically the virus in those two fractions was able to create holes or pores in this membrane barrier. On day 3, the significance went away, and on day 5, the significance came back again for fractions A and C, but not for fraction E. So in conclusion, HIV-infected T cells release various sizes of extracellular particles that contain infectious HIV, excuse me. And we observed that the fractionated large extracellular particles are positive for LAMP1, SE3, REF5, and REF7. These are typical amphysome markers, but mainly LAMP1, large extracellular vesicles contain viral RNA, including genomic envelope, and they are also infectious. Meanwhile, the small HIV was present in fraction E, which is less than 50 nanometers and can pass through 50 nanometer filters. It can be blocked by all HIV-specific broadly neutralizing envelope, broadly neutralizing antibodies. NRTIs did not interfere with the production and the release of the small virus. They have a CD63 phenotype, and we also were able to recover them not just from T cell lines, but also from acutely infected PBMCs. Furthermore, we observed differential in vivo infectivity profile, tissue distribution, and blood-brain barrier damage of HIV contained in the large fraction A, medium fraction C, and small fraction E extracellular particles, which are released from the same J1.1 LAV-infected T cells. And to tie everything together, probably we are facing a model where we have the classic assembly and release of the virus present in fraction C, which is around 130 nanometers. We can potentially have an alternative pathway for the production and release of virus, which is in fraction E, where small extracellular particles and small virus are released together from the infected cells, or alternatively, the small virus and small extracellular particles can exit through late endosomes and amphisomes, which are present in fraction A, and then they are present in the supernatant. And with that, I would like to thank for the guidance and mentorship of Dr. Fataka Shanchi and Dr. Victoria Polonis, and to our collaborators from Touro University, Florida International University, and the support of MHRP leadership. And with that, we are ready to take questions. Thank you.
Thank you very much, Sebastian and Anastasia. What I'd like to do is, if you don't mind, please unmute yourself and ask the question, because some of the stuff that's coming in the chats are not, I don't see all the questions, so it would be nice to just unmute and ask, if you don't mind. Okay.
Hi, this is Lynn Pulliam. I was just wondering about the decrease in size of the neurospheres. This is the first lecture. Anastasia, was there a decrease in size in maybe indicating a loss of cells, infection or death? Because I noticed you saw that the astrocytes and I think even the microglia neurons were on the outside.
Yes, yeah. So very good question. Visibly, we did not see any differences at 3 months. However, we are now keeping them for longer to more better replicate the chronic infection. And now at 9 to 12 months, we are seeing a change in the cell types and composition. And we're actually currently performing a flow to get to the bottom of it.
Okay, I'm just thinking you might want to even just check the size.
Yeah.
Like a, loss of kind of like in the brain when you lose neurons. Also, I was kind of interested in the tetraspanins from the EVs, and this might be Sebastian. CD9 versus 63, because I've always thought of the CD63 EVs coming from macrophages.
Well, we worked only with T cells, so
I know, and
they are present.
it's present, but I'm
They're
just curious
omnipresent.
about that. Yeah. I was just curious because when I do macrophage EVs, they're mainly, CD63. So I was just curious about that. Thank you.
Thank you.
Hi, Anastasia. Great work. So I am little curious about your humanized mice study design. So what is the study design and what is the route of administration of the antiretroviral therapy and the duration? Because in your data we didn't see much suppression of the viremia even in the control animals.
So
Yeah.
it was intraperitoneal injection for the CR. It was treated 3 weeks or 3 times a week over 2 weeks for the CR. And there were up to 3 log drop of the on RNA, but there was not, I agree, a complete suppression.
Okay, thank you.
Thank you. Did that answer your question?
Yeah, okay.
Hi, this is Jack. I have a question for Anastasia and Sebastian. So first, wonderful presentations. I have a question about the decrease of EV production was kind of subtle compared to no cannabinoids, yet you attribute all the effects to that. Oh, well, maybe I got it wrong, but you attribute a lot of the effects to the EV suppression effect. So do you have any comments on that?
Yeah, I think that is a very good question. So I agree. In this case, we did see a more subtle version of a decrease in the 100K, but we've seen it with previous work as well. So it's been a trend that is continuously happening. And then with the escort dependent EVs, I think it's important because those are highly regulated. So even though it is a subtle decrease, it is something that I think is intentional. I don't know if that's the right word, but intentional within the cell. But I do think the other piece is, the decrease in the viral RNA within the EVs and the pro-inflammatory cytokines are not necessarily escort dependent. Those might be going through another outlet, and we're exploring that currently.
Thank you. That makes sense. And for Sebastian, I saw a snippet of the TEM images of the fraction E, but it was a little blurry, and I was wondering, did you capture like the actual HIV capsid or structure like that under EM?
We did capture, and it's in the article, like smaller particles, but we are working on it, having a better resolution TM imaging at this time. That will be the follow-up study and perhaps some cryo-EM. They are under 50, so I'm not stating that the virus is 18 nanometers, but we are stating that with this method we enrich for particles in the 18 nanometers, but certainly they're under 50. So where are they on the spectrum in that fraction? It's a mixture of particles. It's still a question that we actively seek.
I see. And do you think that they're still vesicular, or they could be particles like protein aggregates with RNA complex?
I think it's all the above, to be short and brief. So it's a mixture of everything.
Okay.
That's why we need better imaging indeed, and probably we need to do phenotyping imaging at single particle resolution with cryo-EM.
Okay, that makes sense. Thank you so much.
You're welcome.
Any other questions, comments? Okay, if not, Carolyn, I guess I hand it over to you. Thank you very much.
Yes. Thank you. Thank you so much.
I have
And
one
thank you to our speakers. Oh, does someone have a question?
Yeah, sorry, I'm late.
Oh, go ahead, please.
I just wanted to know, because we use high-speed ultracentrifugation for precipitating EV, right? So for isolating EV. So with this high-speed ultracentrifugation can also result in other non-EV particles to precipitate, right? So leading to possible contamination together with EV preparation. So when and also it may also promote like non-physiological or artificial association between EVs and those cytokines, soluble proteins that you mentioned earlier. So I wanted to know like what methods we use for purifying the EVs so that we don't take in account for those contamination.
So that is a loaded question. There are a variety of different EV isolation methods. Of course, each of them have their pros and cons. For the ultracentrifugation, yes, there can be some artifacts that you see. However, I think if it is something that you can see not just with the ultracentrifugation, but also similarly with the size exclusion chromatography or the SEC columns, then it's something that is at least trending that seems less likely to be an artifact and more likely to be what is clearly there. And then I also, for Sebastian, he can answer this better, but with the images as well, you can see that EV structure that you're looking for.
Oh.
But I do think it is definitely something that when looking at any of these results, you do have to take that into consideration.
Hi, thank you.
Yeah, thank you.
So, Sebastian, do you want to say something? Because I'd like to add a comment here.
Well, my comment, it's only MySEF 2023.
That's the paper we're talking about.
That's the MySEF. I mean, not our paper too, indeed, but just generally speaking, because it was a very general question, depending what one wants to ask. So we did two different methods with centrifugation to isolate the small virus, right? And we did the filtration. Both of them, they have their pros and cons, and both of them contain other parts than just the virus.
Yeah.
I think the best approach and the prudent approach from every in whatever case one would take, it's to prove the same point with two different methods.
Yeah. I'd like to just add a footnote in here. And I think if you go back and look at our initial paper, 2013 paper, up to now, we've published about 12 or 14 papers related to this topic. And every time we're publishing, we're doing much more refinement and improvement. And today so far we've settled on 4 methods. So there's the ultracentrifugation, and then there's the iodixinol gradient, then there's filtration, sizing column basically, and then there is the filters that Sebastian showed you. And I think if the data reproduces in between these different methods and in combination of these methods, then it could be, that could be real. The other point is, the bigger point, I think, is that we've made the claim since 2013 that what we're seeing at least in vitro, in humanized mice, in primary cell infection, and also in patient samples, is not full virus. In other words, we're seeing body parts of the virus in EVs, and therefore, and there are certain criteria that you could call as a virus, right? You have to have 2 genomes, you have to have tRNA, you have to have RT. So there's a whole criteria that one should have in order to call it a virus. And we don't see that. We see 95% of the material in many of these different domains we're touching, in vitro or in humanized mice or even patient samples, that we don't see virus, we see body parts. And I think that adds complexity to not only diagnostics, but also treatment. And as we dig deep into this treatment, into this characterization, we're realizing that not only there are body parts of the virus, but now there are viruses that are potentially smaller. And we're trying to figure out how is that possible? What is the origin of these viruses? And they clearly are coming out at different stages of maybe cell cycle, or maybe they're coming from different origins, from ER versus membrane. So that's a possibility. And then I just see a comment from Amanda that they may be cooperating in infection. I think, Amanda, you're absolutely correct, because when supposedly the virus is released from the cell, it's never just a virus. It is a bunch of things, including EVs, that are released with this virus. So these EVs are essentially a decoy for the innate immune molecules within the neighboring cells? Or what do they do to decrease or increase the level of the cytokines in the neighbor? All of that remains to be seen. We all have to address this at some point. But you're right that it could be that if it was just pure virus coming out, it may not survive. Whereas if it comes out with these EVs or RMP complexes, now maybe it's removing the defense mechanism in the neighboring cells. And it is only then that the virus can enter and really go through RT and integration and replication. At least that's our hypothesis at this point. And yes, we've used protease inhibitors, and that's a can of worms. We'll have the data for you next time, but I don't know the complete answer at this point, and I'll just leave it at that. Thank you.
I was going to say, I did see one more comment about the dose of the cannabinoids. So it was 5 nanomolars for our cell models, and then it was 1 milligram per kilogram for our mice model.
Thank you.
Okay. Thank you, everybody. Thanks so much to our speakers. Just to let you know, we do not have a seminar for the next 2 weeks. Next week is SFN, and then the week after is Thanksgiving. So we'll meet again on Wednesday, December 3rd. Yoelvis Garcia Mesa will present Adenosine, Guardian of the Brain, Possible Role in Protecting from HIV Neuropathogenesis. So we'll see you then. Thank you.
Thank you.
Thank you.
Thank you. Bye.
Bye.